Good afternoon, ladies and gentlemen. Topics of today's lecture is infection, and infectious diseases. In the slide you see here, you see the interacting triad of causative factors of infection. All the factors of infection, influence to the infection, are subdivided to three elements. And these three elements establish if they be shown by that figure, maybe subdivided in vertices of the triangle. The upper vertex is named agent. Agent, this is a microbe causing disease. Among these microbes, properties of these microbes are important. Virulence of microbes, this is a measure of the capacity of causing damage of human cells. Virulence, the capacity of causing disease. Infectivity. Infectivity is a pathogen's capacity for horizontal transmission of pathogen. Horizontal transmission, this is from one adult person to other adult person. Addictive qualities, also qualities of microbes important for causing of the diseases. What addictive qualities, for example, are integration of host genetics and the gut microorganisms. It means host genetic mechanisms in the interaction to the microbiome. This is a total number of the microbes that lives in the certain habitat of the human body, for example, in the gut, and other properties. Exception for agent infection may be developed only if host is present. Host, this is a human being or animal susceptible for the agent causing disease. Host characteristics that I have related are genetic susceptibility to the certain agent causing disease, resiliency, nutritional status, motivation. But nutritional status of the host is very important because starvation usually stimulates development of the disease because host again becomes weakened and microbes they consume the situation for causing of the disease. And third element of this triangle is the environment. Agent microbe interact with the host in the certain environment, and some environmental characteristics are very important to understand what is the infectious disease and would this disease be developed or not. Among environmental factors are public health, sanitation, social context, availability of healthcare, and so on. But public health sanitation. But now we know that we live now in the conditions of the pandemic, of the coronavirus infection. But public health sanitation may minimize chances of the coronaviruses to be spread in the human host population because the mask wearing, vaccination, with these things, the suited to public health sanitation and availability of health care, these things may influence the spreading of the disease. We know that in the China, for example, percent of the infected people by the coronavirus infection is much lower than in the Europe, the United States, or Russia, because the public health sanitation measures realized in the China are most cruel and effective than what sanitation, health sanitation that may be achieved in the United States or Europe, dependent on the different traditions of people, traditions of nations. So today we'll say about what these things. Agent, we briefly characterize these things, and very briefly host. Infection and disease definitions. Infection is a lodgement and multiplication of the parasitic microbe within or on the host's body, within or on. Infection process involves interaction between the human and animals from one side, named the host, and the infected microorganisms from other side, named the parasites. It means infection process is the process of interaction of humans or animal hosts. There are organisms that are infected by microbes and the interaction to microbes capable of causing disease. It means infection, infectious disease can consist of humans only or animals only. Only hosts are not enough to cause the infection. To cause infection, causative agent of this infection is important. Bacteria, virus, or fungus, or other. And only in the interaction, infection may be realized. It may result in infectious diseases. Infectious disease, this is one variant of the development of the infectious process. Infectious disease may be developed after beginning of the interaction of hosts and microbes, or may not. It means infection process without symptoms, but without lesions, without feeling of the pain or other signs of the disease. Infectious process may occur, but this process couldn't be named as a disease because disease, this is an infection associated to damaging of the host tissues, affecting the host functioning. If the infection disrupts the normal function of the host, the disease occurs, infectious disease. So infectious disease is the type of infection in which the clinical manifestation of the particular infectious disease are present. It means disease, this is a type of infection associated with clinical manifestations. For example, in this photograph you see skin lesions and mucous membrane lesions of the lips caused by herpes virus infection. Spreading of the infectious agents in the population of hosts associated to causing of clinical manifestation of disease in some of these infected hosts may be explained by the so-called iceberg concept of infectious disease. This concept declares that only upper part of the iceberg, the tip of the iceberg, is manifested by clinical diseases. Lower part of the iceberg, larger part, submerged portion of iceberg, this is a situation where hosts may be infected, but clinical manifestations of the hosts are not visible, and all the iceberg may be considered as the massive of the infected host, for example, people. Only some of that have the infectious disease demonstrated by symptoms, and majority of the hosts, they are hidden because the hidden form, hidden burden of the disease, because they infected but don't develop symptoms and becomes invisible for the other people. So what in the green field, iceberg concept of infectious disease in population is shown by the pyramid. Upper part of the pyramid, this is a tip of the iceberg, of the black color, the death of patients infected by the certain pathogen. Lower you see more number of patients with severe disease, but they survive. below this severe disease, you see the next part of the pyramid, mild illness, where many people are affected, but the symptoms not very intensive, and red and green elements of this pyramid, they fit for this submerged portion of the iceberg, because with this red sector of the pyramid, this infection without clinical illness, without manifestation, and green, they are people exposed to the infectious agent without development of the infection, without development of the visible infection. They may be, for example, carriers. It means that only low part of the patients demonstrate severe symptoms of disease and may die. Much more patients have mild disease, no clinical manifestations, or so-called carriers that don't have symptoms but may spread infection in other people. Infection, an infectious process, may be shown as a chain of events. This chain consists of the 6 elements. Element first consists of germs. They are microbes causing the disease, among them bacteria, viruses, parasites. These germs, to spread in the population, this microorganisms, may and they must infect so-called reservoir. Reservoir for these hosts are people, animals, food, soil, water, all the things that may be contaminated by the germs. Contaminated. No, in our case, most important for us, the animals and humans. They may serve as a reservoir of infection. The third element, path from reservoir. From this reservoir, microbe may be transmitted to the some people or some animals, and these people become infected through passage of microbes from reservoir to through the portal of exit of the disease. These portals of exit may be mouth, vomit of saliva of infected peoples that are members of reservoir population. These portals of entry may be cuts in the skin, when bacterium enters to the blood, and other things, due to the stew and eating of the food. So germs may enter to the susceptible people through the portal of exit. In portal, gastrointestinal tract, for example, blood. After that, the third element, we have to say about transmission of infectious agent. What this portal of exit, people that are shown here, they are patients number 1 of the first category. From these patients, through the vomiting, stool, dirty skin, other things, microbes may enter to the bodies of other people. And the transmission of this of the reservoir first patients to the other people is characterized by the mode of transmission. This mode of transmission, what this is the yellow field. Boy, for example, releases microbes with the saliva. If the saliva is inhaled to the next person may acquire the disease through the contact towards skin or the saliva of the infected person, and germs get around by the resolution of mode transmission. This modes of transmission may be droplets, contact, Transmission may be associated to the contact of hands, toys, sand, and so on. Fourth, mode of transmission. Fifth element of this chain is named path of entry. Path of entry. So, how germs getting portal of entry? Portal of entry, there are organs or tissues that may be infected and colonized by microorganisms. This portal of entry may be mouth, cuts in the skin, eyes. After infection of these organs of the humans develop disease, and we come to the susceptible hosts. To the hosts, they in the moment of contact to this infected people where portal of entry becomes infected, and some of these susceptible hosts marked by the number 6 becomes infected. The second population of infected people, not the first reservoir, but second, and they are sources of transfer of disease in the form of the microbes. They release these microbes, and some of these people establish the more number of people in the reservoir, and the cycle repeatedly continued. So, chain of infection consists of the pathogenic organism. Here, virus is shown. Reservoir. The are organs of the people or animals that carry this pathogen in their organisms. The third element of the chain, exit portal. Exit portal, the virus exists in the body in infected droplets spread into the air by cough, sneeze. It means with this cough and sneeze, responsible for exit of the microbes to the other people through the transmission. It means microbe is released from the nose or mouth after the droplets or dirty skin contacts to the different things, to the bowl, utensil, and other things that may be infected after that, and transmission through these fomites causing entry of the microbes to the portal of entry to the organs. They are new people. Some of them becomes infected and surface the new hosts for this parasite, and they continuously release this pathogen to the environment, and all the circle is continued. Microorganisms that cause disease. What are the characteristics and terms characterizing this microorganism? Depending on the relationship with their hosts, microorganisms can be classified as saprophytes and parasites. All the variety of microbes may be subdivided to a large group of saprophytes and a large group of parasites. Saprophytes. Saprophytes, they are free-living microorganisms. Free-living, it means they may survive in the environment without infecting humans or animals. Saprophytes are generally incapable of growing and multiplying in living tissues of the host. Usually, as free-living, they live in the objects of environment without surviving in the human tissues or the surface of human tissues. Some saprophytic organisms, such as the Bacillus subtilis, gram-positive spore microbe, but some Bacillus subtilis microbes may infect so-called immunocompromised hosts. Immunocompromitation, this is a deep lowering of the immune responses in the body of this patient. With this lowering of immune response may be caused by starvation, by the other disease, for example, human immunodeficiency virus infection, by other causes. It means if people are immunocompromised, saprophytes, some of them may colonize their tissues and live in these tissues. After these conditions, these microorganisms may cause infectious diseases called opportunistic infections. In the photograph here, you see soft palate and larynx of the patient with the candidiasis. You see red inflamed mucus of the patient's mouth, of the palate, and again this red color you see white dots. By dots there are layers and colonies of the Candida albicans. This is a yeast fungus. Opportunistic infection caused by this fungi usually may develop only if immune system of these patients is broken, is weakened. For example, in the HIV patients, this may occur. Or if patients have the severe metabolic disorders, for example, had developed diabetes mellitus. Parasites, second group of the organisms. In the contrast to saprophytes, parasites are capable of growing on living tissues of the host, including persons whose natural resistance is not reduced. It means parasites may live on living tissues of hosts, even in case of the normal function and normal status of the immune system of these hosts. Parasites are different. They have different capacity of the colonizing of the tissues of healthy people. Due to this, parasites may be subclassified for the group of commensals, facultative pathogens, and frank pathogens. Commensals are microorganisms that live in a host without causing any damage to the host. These are normal flora microorganisms, for example, Escherichia coli. Escherichia coli lives in our large intestine and other habitats without causing disease. They are parasites because may live in the living tissues, but commensals because in people with normal immune status, commensals can cause disease. The next group, more virulent. They are named facultative pathogens. Facultative pathogens are the commensals which can produce disease in the host when the host resistance is lowered. Facultative pathogens, they need to cause disease need lowered host resistance. But many fungi have these things, and we know that these facultative pathogens may cause opportunistic diseases. And last group, frank pathogens, are microorganisms capable of producing diseases in the hosts with a normal immune system. For this, they should have properties of enter the body and disrupt the state of health. Some are highly pathogenic and often cause disease. For example, Shigella boidae, Shigella zone, Shigella dysentery, the species of Shigella causing shigellosis. Shigellosis clinically is a colitis, ulcerative colitis, when ulcers are formed on the mucous membranes of the large intestine. When characterizing capacities, different capacities of different markers of causing disease, we have to comment such terms as the pathogenicity and virulence. Pathogenicity and virulence, they are characteristics of microbes that relies in the causing of the disease, the capacity of causing disease, but they have clarity. Pathogenicity is the capacity of microbial species to produce disease. What is the critically important word? Microbial species. It means pathogenicity is characteristic of species. It means total collector of all the strains of the given species in the world. It depends on the ability to invade a host, multiply there, and avoid being damaged by host defenses. Virulence. Virulence refers to the ability of the microbial strain to produce disease. It means virulence is not the property of the species. Virulence is the property of the certain microbial strain to produce disease. It's a quantitative measure of pathogenicity. It means virulence is the measure of pathogenicity, quantitative, it may be measured, and is measured by the number of organisms required to cause disease. It means virulence is measured as the number of microbes required causing disease in the host. The virulence of a strain is not constant and may undergo spontaneous or induced variations. It means with this virulence, the quantitative characteristic of pathogenicity of the causing of the disease may be changed, and the changing may be directed for the enhancement of the virulence or lowering of the virulence. Lowering of the virulence is named attenuation. Attenuation is a reduction of virulence that can be achieved by cultivation of the certain virulent strain under the unfavorable conditions. It means if you will isolate a certain state of microbes, of virulent microbes, you may attenuate them, reducing the virus by the culturing under unfavorable conditions. What are these conditions? Passage of the microbes through unsusceptible hosts. For example, if we take the pathogen of the flesh-eating organisms, for example, so causative agent of the madness, mad dog may bite humans and transmit this disease, the Lyssa viruses, or rabies viruses. It means causative agent of rabies, if is cultured during long time in the unsusceptible hosts, for example in rabbits, finally after many weeks may be changed, and this changed attenuated strain of the rabies virus would be attenuated and may be used as a vaccine. Low pathogenic virus that causes the intensive immune response, protecting against wild pathogen with a high virulence. Second variant of the attenuation, repeated cultures in artificial media. So you may culture microbes long time in the pure artificial media, and some bacteria in this case lose their virulence. Third, growth under high temperature or in the presence of weak antiseptics. With high temperature, more than 40 Celsius, and presence of antiseptics in growth media may also cause attenuation. It means weakening of the virulence of the states that undergo this attenuation. They are things associated to the lowering of the virulence. What about enhancement of the virulence? Virulence of the certain strains may be increased by animal passages. For example, transfer of the pathogen through animals of susceptible species. It means passages through the new hosts in some conditions may cause attenuation, in other may cause enhancement of the virulence. So, what in this slide you see definition, measurement, and use in the explaining the harmfulness of the terms virulence and pathogenicity. Definition. Virulence is the measurement of the ability to cause the disease in the host organism. This is a property virulence of strains. Pathogenicity. Pathogenicity is the ability to cause disease in host organism, but ability of the species, not strains, but of the species of microorganism. It means we can say that Shigella is a pathogenic microorganism. It means that all strains of Shigella will cause the Shigellosis. The second option, measurement, virulence, can be represented by the quantitative measure. It means virulence may be measured quantitatively. Pathogenicity. Quantitative measure is not quantitative measure of the pathogenicity may be related only with the measurement of the virulence. It means virulence is a quantitative characteristic, and the pathogenicity is qualitative measure, not the quantitative, of the capacity to cause the disease. And used in explaining the harmfulness. Virulence can be used to express the degree of harmfulness of a pathogen. Pathogenicity, not much suitable to explain the degree of harmfulness of a pathogen. What we say about species, там Shigella dysentery, all the strains of the species are harmful. But if we'll compare two different strains of the Shigella dysentery, one strain may be more harmful, second is less harmful, and this is a difference that is explained associated to the property named virulence, not the pathogenicity. Now about measuring of the harmfulness, measuring of the virulence of microorganisms. Virulence or virulence of microorganisms. This is quantitative characteristic capacity of causing disease of the strains. Here you see 1, 2, 3, 4 indices that were introduced in the measurement of the virulence of the microbes. First, LD50. This is a lethal dose 50%. This is a 50% lethal dose in the number of microorganisms needed to kill half the infected hosts, laboratory animals. It means to measure this lethal dose 50, we have to take, for example, 100 mice. If all of these mice would be infected by the 50% lethal dose of the microbe, Staphylococcus aureus, 50 LD50 would be when half of these mice will die, half will survive. The next index is minimum lethal dose. Minimum lethal dose is the minimum number of the microorganism. Also, LD50 is the minimum, and MLD also minimum of the microorganism required to kill 95% of the animal tested under the standard conditions. It means if we will infect by the certain Streptococcus animals, for example, guinea pigs, 100 guinea pigs, if almost all of them will die, but some few, some 3 or 5 animals will survive, dose causing this effect would be named minimum lethal dose. Usually minimum lethal dose is higher than LD50 because number of the killed hosts will be different. 50 half infected mice in the LD50 and almost all 95% in case of use of minimum lethal dose. ID50, this is 50% infectious dose. This is the number of microorganisms needed to cause infection in half the hosts. ID50, this is a 50% infectious dose causing not the death, but clinical manifestations. It means that number of survivors among infected animals would be higher than 50%. But 50%, half of them will demonstrate symptoms, for example, diarrhea. And last one, MID, minimum infectious dose. This is minimum infectious dose, and the minimum number of the microbe is the minimum number of the microbe required to produce clinical evidence of the infection. It means minimum infecting dose is the minimum number of microbes required to produce clinical evidence of the disease in 95% of the infected animals. So the infectious dose, minimum infectious dose of Shigella is less than 100 organisms. It means that 95 cells of the Shigella, when entered to the human body, may cause the disease development. Minimum infectious dose of the Salmonella is on the order of 100,000 organisms. It means if you will inoculate to the animal 50,000 organisms of Salmonella, this may be not enough to cause the disease, but 100,000 and more is enough. What the organism is more virulent, Shigella or Salmonella? Of course, Shigella, because dose of Shigella that guarantee causing of disease in the host is only 100 cells of the Shigella bacteria to cause the analogous effect. In case of inoculation of Salmonella, we need to inoculate 100,000 organisms. It means virulence of the Shigella is 1,000 times higher than virulence of Salmonella. How bacteria cause disease? Bacteria cause disease because they have so-called virulence factors. Virulence factors are the organelles of the microbes, enzymes released by the microbes, and other organelles or properties of the microbes involved in the development of the disease. This is a weapon of the microbes. Virulence factors of bacteria are structural or physiological characteristics that help organisms to cause disease. Virulence factors are classified for adhesive factors or adhesins. This is possible for sticking of bacteria to the human cells. Second group, colonization factors. They are factors that help not the single bacteria, but colony of the bacteria survive in a certain part of the human body. Third group of virulence factors, invasiveness factors. Invasiveness factors mostly are enzymes that disrupt human cells or substance between these cells and help bacteria to spread in the human body. Invasiveness, this is a capacity to be spread inside of the host organism. And last one is aggressiveness factors. Aggressiveness factors are structural compounds or substances of microaggressive bacteria that help these bacteria be resistant to the human cells and the human immune system. It means aggressiveness of microbes. This is capacity to bypass immune response mechanisms of humans. Aggressiveness factors protect bacteria from attack of the human leukocytes or antibodies or other immune factors. Adhesins or adherence factors. Adhesins and adherence, adhesiveness and adherence is the ability of the bacteria to attack a host cell surface. It's a specific reaction between the cell surface receptors and bacterial adhesins. Adhesins are adhesive structures of the surface of bacteria. Among them, fimbria or pili and capsules or slimy layer. Not all the pili are adhesins of the bacteria, but fimbria or simple pili pili of the bacteria, for example, of the Neisseria gonorrhea shown here, mediate the attachment of this bacteria to the urinary tract epithelium. Here in the photograph you see two cells attached one to other, Neisseria gonorrhea adipococci, and every cell is surrounded by fibers of the filament fibers, and these fibers consist of protein, and these fibers are named pili, adhesive pili. These fibers attach bacteria to the epithelial cells of the urinary tract, establishing gonorrhea. Second adhesiveness factor is capsule, the slime layer. What in the photograph, the right lower part, you see Staphylococcus, the gram-positive organisms, that are surrounded by the white capsule. This capsule protects this bacteria from phagocytosis, and in this way serves as the aggressiveness factor. From other side, the capsule material surrounding bacterial cells attach this bacteria to the surfaces of the human cells. Slime layer, this is a very large capsule that serves as a common shell surrounding group of bacteria. Slime layer is not the individual capsule of one cell, but this is a collective capsule material surrounding group of cells. Slime layer of Staphylococcus epidermidis mediates the attachment of the bacteria to the endothelium of heart valves. With Staphylococcus epidermidis may migrate in the blood, may attach and form colonies in the endothelial cells of the heart valves. And in this seeking absorption of the staphylococcal cells to the endothelial cells of humans, very important role is the role of the capsules, polysaccharide capsules of these bacteria. In this slide, you see mechanisms of adhesions by pathogens. You see three different situations, upper situation. Host cell becomes colonized by bacteria due to the bacteria fibria. You see filamentous fibria, they attach themselves to the chemical groups on the surface of host cell. The bacteria becomes absorbed to the cell. This is a fibria. Second, intermediate situation is named capsule. You see that host cell of the yellow color is coated by the green material, and bacteria situated inside of this layer of the green material. This is a biofilm on the surface of the host cell, and the bacterial cells are surrounded by capsules. What capsules that are shown here, what for example you see 1, 2, 3 bacterial cells that are packed in the one element of the capsule material. This type of capsules is named slime layer. And the spikes, the option C, spikes, we see that on the surface of the human cells are absorbed viruses that have spikes. They are envelope viruses because spikes usually integrate into the envelope. These spikes interact with the receptors of human cell and help this virus to enter to the cytoplasm of the cells, mediating penetration of host cell by the virus. Colonization. Second capacity of bacteria that deals with the capacity of causing disease. Colonization refers to the growth of microbes on the epithelial surface or other host tissue. Refers to the growth of microbes, not only attachment, but attachment and the fuse attachment that further is shaded to growth of bacteria and colonizing, multiplying of the bacteria on the surface of the human tissues, covering these tissues. For colonization to occur after adherence, the microbe must survive and reproduce despite host defense mechanisms. In the illustration, the lower part of the slide, what is the A part of this illustration, you see three steps of the colonization of surface of the human epithelium. First, three bacteria are shown of the green color, and one of them becomes attached to the surface of the epithelial cell. After that, this attached bacteria replicates, producing progeny. If this progeny is capable of the coating of the surface of human epithelial cell, if this young colony is not detached and not killed, we say that what beginning from this moment virulence activation is happening. It means activation of factors that will protect killing of bacteria forming colony on the surface of the human epithelium. And the third step of this process is colonization. It means not monolayer bacteria only shown here, but you see many layers that form the biofilm forming colony on the surface of this epithelial line that you see in the slide. In the right upper part of the slide, you see colorization of the surface of the enamel of the teeth. Enamel of the teeth is shown as the gray blue material by the bacteria. Bacteria form a slime layer, and this slime layer here is shown as the yellow gray material. This slime layer consists of the bacterial polysaccharides that protect them and stick them, touch the surface of the enamel, and bacteria that are hidden in the depth of this slime layer. In this slide, you see multi-layered structure of the biofilm that is formed due to the colonization of the tooth surface by many bacteria that normally live in the mouth. Bacteria growing on tooth enamel shown, to which they initially adhere by means of the slime layer. What slime layer of this bacteria shown by what this layer of the green color. This is a slime layer. Here, slime layer shown only the material that help bacteria to be attached to the surface of the enamel. Well, this slime of the bacteria, for example, Streptococcus sanguis, Streptococcus orale, so Streptococcus gordoni. These bacteria, they additionally accumulate their polysaccharides, increasing slime layer, and these bacteria, after attaching to the surface of the tooth, interact to receptors of other bacteria that form the second layer of the biofilm that coats the human teeth. What is the second layer? Capsules is present, Propionibacteria, Villanella, or this red gram-negative bacteria, Privotella organisms, Echinella, Actinomycetes. In the turn, these organisms also produce layers that are touchable for bacteria of the third line that are attached to this second line bacteria. Among the third type of bacteria are fusobacteria that are shown as the long cells that form the fur-like line on the surface of the teeth that also absorb to them new bacteria. For example, Treponema denticova that is shown here. So finally, you see complex consists of the 1, 2, 3, and 4 layers of microorganisms. All these microorganisms occur on the surface of the tooth and are integrated in the depth of the slime layer. Really, slime layer is larger than only this green layer intimately shaded to the surface of tooth surface. This slime layer occurs in every part of this colony, causing the common protective biofilm that contains different types of microbes in the depth and protects them from killing by the bactericidal factors of the saliva and antiseptics. Invasiveness. So before we said about adhesiveness factors and colonization factors. Adhesiveness, this is an individual capacity of one cell to be attached to the human cells in the habitat of the human body. Colonization is the capacity of not one but many cells that are descendants of these first cells that were attached to the cell, to the human cell surface. It means colonization is the cooperative property of the many bacteria. Third virulence factor is named invasiveness. Invasiveness is the ability of a pathogen to spread in the host tissue due to the secretion of enzymes. This is a capacity to spread in the host. What are the enzymes? For example, hyaluronidase and fibrinolysin. Hyaluronidase secreted by Streptococci. Streptococci in the intermediate part of the slide are shown with the yellow balls. Streptococci. So hyaluronidase secreted by Streptococci dissolves the cementum that holds together the epithelial cells. What cementum? This green material between neighboring epithelial cells. This cementum holds together the epithelial cells that lies gastrointestinal tissue. If bacteria produce hyaluronidase, dissolve this connective tissue, this cementum, neighboring bacteria becomes detached one from other, and bacteria and bacterial products may come inside of this space between cells, and finally may go to the space between epithelial cells and basal membrane of these cells. And after that, Staphylococcus dissolving the connective tissue below the epithelial cells becomes capable of colonizing of the deep parts of the human tissues. The second variant, fibrinolysin, is also streptococcal enzyme, for example, Streptococcus pyogenes. Fibrinolysin is also named the streptokinase of Streptococcus pyogenes, dissolves the fibrin clot, and bacteria trapped within a clot can free themselves and spread in the body. But in the lower part of the slide, you see three phases of the colonization of bacteria, colonization of tissues by bacteria. First step, through the blood vessels, streptococci enter to the perivascular space. We see here black round cells of the streptococci and green dots, the enzymes secreted by bacteria. These enzymes cause the formation of the clot. It means clotting of the human protein surrounding the bacteria. Formation of the clot serves as the protective mechanism of preventing of the destroying of the bacteria by the immune factors of human organism, by the phagocytes. But later, when number of the bacteria inside of this class becomes high, and the strength of this colony of bacteria becomes high, bacteria dissolve, producing red substances, this is a fibrinolysin, dissolve the fibrin elements of this clot, liquefy them, and bacteria after that may spread themselves from this focus of infection to the neighboring tissues. For example, to the perivascular space and in the vessels. In this case, you see two instruments of the survival of the bacteria. First, formation of blood clot caused by bacterial coagulase. After, liquefying of this clot with removing of bacteria to the deeper tissues and blood caused by streptokinase. In this case, streptokinase serves as an invasiveness factor because facilitates spreading of bacteria in the human body. In this slide, you see how Helicobacter pylori invade tissues in the stomach. Helicobacter pylori shown in the green bacterium, the three flagella, to cause damaging of the human cells must intimately contact to the epithelial cells of the duodenum and the stomach. But epithelial cells produce mucin, and in the presence of high concentration of the hydrogen protons, it means the acidic environment, mucin for the sponge form gel-like material that prevents direct contact of the bacteria to the epithelial cells. But these bacteria produce urease. Urease, this is an enzyme, bacterial urease, that neutralizes the stomach acid, neutralizes acid. pH becomes higher in this point. And this causes the mucin liquefaction. Mucin becomes liquefied in the non-acidic conditions. And bacteria move to the epithelium and make direct contact with this epithelium after that, destroying these epithelial cells and starting of causing ulcer. Aggressiveness factors. Aggressiveness factors protect bacteria from phagocytosis and other immune reactions. Phagocytosis is other immune reactions. For example, coagulase of Streptococcus pyogenes, coagulase, shown here the blue dots, accelerate the formation of the fibrin clot, well, this is a fibrin clot, from fibrinogen. Coagulase makes fibrinogen to form fibrin polymer, and this polymer serves as the net protecting clot from the destroyed by the human or bacterial enzymes. It means coagulase of Streptococcus pyogenes accelerate the formation of the fibrin clot from fibrinogen. This clot may protect the bacteria from phagocytosis in the period of infection when number of bacteria is low. Bacteria replicated in the future, their number would be enough to attack human immune system. But before this, coagulase, via production of clot, protects bacteria that form colony. In the future, this fibrinogen would be dissolved by the second enzyme of the Streptococcus pyogenes, that is streptokinase. Streptokinase is invasiveness factor. We talked about this. That dissolves fibrin clot, and bacteria trapped within a clot can free cells and spread in the body. What streptokinase is shown by the red dots. This is an invasiveness factor. Coagulase, the aggressiveness factor, shown as the many blue dots here. So, streptococci have at least two different instruments suited to invasiveness and adhesiveness of these cells that help them in their colonizing and surviving the human tissues. Invasiveness staphylococcal factors, the hyaluronidase, and collagenase. Hyaluronidase dissolves the cementum between neighboring epithelial cells. Collagenase destroys collagen. From that is formed the basal membrane. Both these enzymes cause passage of the microbes through the space between two separated cells and through the hole made by the coagulase in the by the collagenase in the collagen tissue, and result spreading of the microbes in the human tissues. If bacteria enter to the blood, they tend to spread in the human body, may pass through bloodstream to the perivascular tissues. Bacteria shown by the gray dots, and the enzyme coagulase is released by this bacteria in this phase of the colonization. Coagulase interact to the fibrinogen and converts fibrinogen to the fibrin, causing polymerization of fibrin. Result, formation of the clot that protects bacteria from the phagocytes. This is a demonstration of aggressiveness factor. And third, right part of the slide. When fibrin clot is not required for protect bacteria because bacteria replicate and achieve very high concentration, the invasiveness factor, that is kinase, start to be secreted by these bacteria. If the number becomes high, they communicate one to other by the so-called autoinductors, and the autoinductors stimulate beginning of the synthesis of streptokinase. Streptokinase or fibrinolysin dissolves the clot and accumulated in this place bacteria from this spread to the human body, for example, by the entry into the bloodstream. Here you see collaboration between invasiveness factors and aggressiveness factors in the different stages of the infectious disease caused by Streptococcus. Of course, very important for aggressiveness factor is the capsule. We said before that capsules are attachment adhesins, of course, but additionally, capsules are aggressiveness factor. Some factors may limit the ability of the host defense mechanisms to operate effectively. Capsule of Streptococcus pneumonia is one of the most important antiphagocytotic factor. It means polysaccharide capsule Streptococcus pneumonia, capsules shown white, seen green, pink background, and some dark round cells inside. This capsule is a powerful antiphagocytotic factor of Streptococcus. In this slide, you see two bacterial cells of Bacillus anthracis, and each the cell is coated by the large capsule, shown green because this green color is a result of the staining of the cells by the fluorescent dye, fluorescein, that when it's radiated by the ultraviolet light, emits the light in the green part of the spectrum. The next group of virulence factors, exception for adhesins, colonization factors, aggressiveness factors, and invasiveness factors, the fifth group of virulence factors are hemolysins. Majority of them are classified as the toxins, if are the proteins, but some of them are not proteins and not classified as the toxins. The action of hemolysin is to lyse erythrocytes. Here you see A and B petri dishes colonized by different microorganisms. Two types of hemolysins were identified on blood agar. Blood agar that demonstrate changing of the hemoglobin if the changing is made by bacteria. A, upper slide on the left. demonstrates so-called alpha hemolysin of the Streptococcus. This is a partially breaking down of hemoglobin. Partially, not completely, destroyed hemoglobin converts color from red to green. And this green color produced by the Streptococcus is one of the differentiation characteristics of these cells. B. Lower petri dish with the red blood cells blood agar demonstrates beta hemolysis. Beta hemolysins. This is a complete breaking down of hemoglobin and leave a clear ring around colonies. You see small colonies. They are surrounded by yellow zones, but they're really zones that have the color of the agar. Red blood cells here are completely broken, and hemoglobin also is completely broken and becomes colorless. With the beta hemolysins, beta hemolysis is a complete breaking of the hemoglobin. Alpha hemolysis is a partial breaking down of the hemoglobin. Very important property of some bacteria that help them to cause disease is intercellular survival of bacteria. Not all, but some bacteria, they possess this intercellular survival. Intercellular survival is important attribute of certain organisms called intercellular pathogens, or intercellular parasites. This intercellular survival protects these organisms from antibodies and neutrophils and other phagocytes that function extracellularly. It means bacteria hide themselves in the depth of the bacterial cytoplasm. Some mechanisms of cellular survival are mechanisms. First, inhibition of the fusion of the phagosome with the lysosome. Now we know that many bacteria, after attachment to the surface of bacteria, becomes engulfed by these human cells, these attached bacteria, and they, after that, are situated in the endosomes or phagosomes, and later lysosomes fuse themselves to these phagosomes or endosomes and inject to these lysosomes enzymes and acidic compounds. This fusion is responsible for contact of the engulfed bacteria to the bactericidal factors. So, one of the mechanisms of the intercellular survival is inhibition of the fusion of the phagosome with lysosome. In this case, bacteria remains in the phagosomes, but then not fused to the lysosomes, and the acidification of the environment surrounding bacteria and presence of enzyme is absent. And this allows the organism to avoid the degradative enzymes. This mechanism is typical for Legionella and Mycobacterium, including Mycobacterium tuberculosis. The second mechanism of the cell survival, inhibition of acidification of the phagosome. This inhibition of acidification of phagosome reduces the activity of the lysosomal degradative enzymes after fusion of lysosome to the phagosome. This is also typical for Mycobacterium and Legionella. Fourth, third mechanism, escape from the phagosome into the cytoplasm before fusion of the phagosome to the lysosome. Where there are no degradative enzymes, it means if this is typical for Listeria and Rickettsia. What you see here, stained red Rickettsia, that accumulate in the cytoplasm and free cytoplasm, not in the phagosomes or endosomes. They are initially engulfed by the phagocytotic mechanism, but after they destroy wall of the lysosome and enter to the cytoplasm directly. And they leave these phagosomes, and in this way protect contact of bacteria to lysosomal enzymes. And final mechanism, resistance of lysosomal action of enzymes. Some bacteria directly protected from digestion by lysosomal enzymes. Example, Mycobacterium lepra and some species of the Salmonella. Very important virulence factors are toxins. Toxins are the highly effective destructive functions, destructive substances of the bacteria and fungi and some other organisms. Toxins, in the contrast to other virulence factors, act in very low concentrations. They are substances that have very high capacity of destroying or causing dysfunction of the human cell physiological and biochemical instruments. Other virulence factors, they cannot usually cause the symptoms of disease, but toxins taken in the chemically pure form may cause one or more symptoms typical for the disease associated to bacteria producing this toxin. It means toxins are poisons of microbes. So, toxins are divided to exotoxins and endotoxins. Let's compare these two different types of the vaccines, the toxins. So, clinical effects first. It means capacity of this substance of causing specific clinical symptoms, specific for the certain disease. Exotoxins are highly specific for each exotoxic disease. For example, Clostridium tetani produced toxin that is named exotoxin, tetanospasmin, that cause the spasm of muscles that can be relaxed after the spasm. In the contrast, endotoxins are non-specific. They cause non-specific effects that are mostly fever and shock. Shock, this is a dramatic fall of the blood pressure that may cause the death. A fever and increasing of the body temperature. It means exotoxins cause the effect specific for the certain disease. Endotoxins not. Common fever and shock in case of the severe disease. Tissue affinities. Exotoxins are specific for certain tissues. For example, toxin of the Clostridium tetani is neurotoxin, affecting synapses. Endotoxins, they are non-specific. They don't have specific capacities to be absorbed to the certain cells. Mode of action. Exotoxins, they are various. Action often enzymatic because they are proteins. Endotoxins, they have no enzymatic action. They induce production of the tumor necrosis factor and interleukin-1 and some other cytokines by immune cells. And this hyperproduction of these pro-inflammatory cytokines causing fever and inflammation. In the contrast to the tetanospasmin, Clostridium tetanus toxin that causing, for example, spastic paralysis without increasing the body temperature and without inflammatory reactions. Exotoxins are proteins and the toxins are lipopolysaccharides. Antigenicity. It means capacity of causing a specific immune response. Exotoxins, highly antigenic. They induce antibody called antitoxins. Endotoxins are purely antigenic. Antibodies may be secreted by the low protective. Neutralization by antibody of the exotoxins. Exotoxins can be neutralized by antibodies very effectively. Endotoxins can be neutralized by antibodies effectively. And antibodies usually don't remove all the symptoms of the endotoxin damaging caused by endotoxins. Vaccines. No, few words about vaccines. A vaccine, this is a component of microbe or genetic construct that calls for component of microbe that is harmless from one side, but can initiate development of specific immune response. It means vaccines are harmless components of bacteria that when is injected to humans or animals, if this animal that is in the vaccine, in this case they initiate synthesis of antibodies and immune response that will protect this person or animal from the wild pathogen. It means microbe of the same type that circulate in the environment. It means exotoxins may be converted to the toxoids. It means if you take exotoxin, pure exotoxin Clostridium tetani, mix this to the low concentrated formaldehyde, heat it at 35 Celsius, and exposed to these conditions during 1 month, resultant product would be vaccine that is not toxoid. This toxoid will consist of the partially chemically modified exotoxin that will lose the toxicity, capacity of damaging of human tissues, but will keep unchanged property that is named immunogenicity, property to induce immune response, antibody synthesis, for example. What about endotoxins? They're not proteins. No vaccine is available. I mean, no vaccine made from endotoxin. Endotoxin can be toxoid. The next characteristics: source of toxins. Endotoxins, exotoxins, typical for gram-positive and gram-negative bacteria. Endotoxins, typical for gram-negative bacteria only. Secreted from cell. Well, this is important characteristic. Exotoxins are named exo, it means external, because they may be secreted from the leaf bacterial cell, easily secreted. Endotoxins can be easily secreted. No, they can be secreted because they are part of the cell wall, the integral part of the cell wall. And endotoxin may be released from bacterium only after death of this bacteria, disruption of the cell wall of this bacteria. It means toxic effect is caused only when cell that is source of this toxin is killed. Gram-positive negative bacteria producing exotoxins survive in the process of the secretion of the toxins. Chemistry. Exotoxins are polypeptides. We remember that they are enzymes mostly, they are proteins. Enzymes are proteins. Endotoxins chemically are lipopolysaccharides. They are not proteins. Heat stability. Exotoxins heat labile and destroy at 60 Celsius during 1 hour. Endotoxins heat stable at the 100 Celsius for about 1 hour. It means boiling of the toxins can destroy them during 10 or 15 minutes. Exotoxins. Location of genes. Genes that code for exotoxins are situated in plasmids or bacteriophages, usually. Endotoxin, genes responsible for synthesis of exotoxins, are localized in the bacterial chromosome. Toxicity, capacity of cause poison effect for exotoxins is high, It means they act in very low concentration, lower than 1 microgram per kilogram. Endotoxins, they low toxic. They have to be used in high concentration to cause toxic effect. And the typical diseases associated to exotoxins and endotoxin. Exotoxins usually associated to such diseases as tetanus, botulism, and diphtheria. Endotoxins associated to meningococcal meningitis, that is named meningococcemia. This is one of the clinical form of this meningococcal meningitis. And sepsis by gram-negative rods, gram-negative rod-shaped bacteria. I have to explain this step. Sepsis, this is a systemic inflammatory reaction that mostly is demonstrated by the changing of the vascular permeability and blood flow on tissues. But if this flow is affected, what usually this situation is may be demonstrated by sepsis. This is a severe disease that frequently causes death. When saying about toxins, bacterial toxins, we have to say about 5 types of the secretion systems. Secretion systems discovered in the gram-negative bacteria. But here in this drawing you see 1, 2, 3, 4, 5, 6 types of channels that help to transport toxins that are shown with here from the cytoplasm through the cell wall of gram-negative bacteria to the extracellular space. So now I say about gram-negative bacteria. So secretion system type 1, type 3, and type 4. Type 1, what this one, 3 and 4, what this in the left part. These secretion systems transport proteins in one step. Transport of proteins, including exotoxins, are what this, and this. In one step, from the cytosol of the cell produces this toxin to the cell surface and out, to the cell surface and out. You see type 1, 3, and 4 consist of the channels that transport proteins to the outside with the consuming of the energy in the form of the adenosine phosphate, here, here, and here. In the case of the type 3 and 4, what type 3 and 4, what's this? Type 3 and 4, secretory systems, the proteins secreted are transported from the bacterial cytoplasm, of bacteria producing toxin, to the target cell cytosol. What this brown material, this is a fragment of the cell membrane of the target cell. It means secretory system type 3 and 4 directly inject transport toxin from the bacteria pathogenic to the target cell. With this plasma membrane shown, demonstrates that type 3 and 4 secretory systems, they looks like the molecular syringes that inject proteins to the directly to the cytoplasm of the target cells. Type 2 and 4 with these red systems, different from the type 1, 3, and 4 because release products from the producing cells were 2 steps. First step and second. First step of secretion associated to transmembrane proteins named CAC, SEC, and TAT. No, second steps are different, but in any case, type 5 and type 2 secretory systems, they release proteins outside without injection to the plasma membrane of the host cell, that is typical for type 3 and type 4 secretion systems. What secretion system type 3 is typical for transport of the toxin of the Yersinia pestis, caused infection of the plague, from body of these cells to the target cells, for example, the endothelial cells of the vessel line, that causes damaging of these endothelial cells. Examples of the endotoxins. What here you see very primitive scheme of the action of the diphtheria exotoxin. Diphtheria, this infectious disease that mostly causes damage of the larynx, but toxins may cause heart defects and kidney defects and cause the death of patients. So, Clostridium, Corynebacterium diphtheria, causative agent of this disease, as the main virulence factor has diphtheria toxin. This is so-called AB exotoxin. In the AB exotoxins, A subunits are directly toxic, causing damaging effect. B subunits are responsible for attaching of the toxin complex to the cell, host cell, and to the transport of the A subunit in the cytosol of the cell. It means diphtheria AB toxin attaches itself by the B portion to the cytoplasm, to the cytoplasmic membrane, irritates the mechanism of the cellular endocytosis. Toxin becomes endocytosed. After that, after the cytosis, B subunits remain associated to the endocytotic lipid membrane. A subunit is detached from B, enters to the cytoplasm, contacts to the elongation factor 2, this specific protein that controls synthesis of the protein in the ribosomes. Ribosomal synthesis of protein is blocked, and when protein synthesis is high, cell dies because can survive when protein synthesis is blocked. This is also scheme of the diphtheria exotoxin activity, but more detailized. So diphtheria toxin really consists of three elements, A, B, and T. B elements stimulates engulfment of this toxic complex to the endocytotic vacuole. After that, T portion of the toxin integrates itself to the lipid wall of the endosome, and A portion after this mechanism becomes transported and released to the cytoplasm. With this confirmation of the structure of the exotoxin is happened when acids are injected to the endosome after fusion of endosome to the lysosome. This fusion is not shown, but you see hydrogen atoms. Hydrogen is a carrier of the acidification that causes conformation of the AB structure of the toxin and the following cutting of the A subunit from B and T elements. B and T elements remains in the endosome. A subunits combine one to other, interact with the elongation factor 2 and causing ribosylation of the elongation factor 2. It means adenosine diphosphate ribose group because attached to the structure of the elongation factor 2, and after that this factor blocks the protein synthesis and cell dies. This is the diphtheria. Second toxin, exotoxin, also exotoxin, Vibrio cholera exotoxin. Vibrio cholera produces AB toxin consisting of two different subunits of A, A1 and U, A2 subunits, and 5 B subunits. Via contact of the B subunit to the receptor of the cell, all this complex is engulfed by the cell. and A1 and A2 portions are dissociated. A1 portion is the presence of the acidic hydrogen atoms, increases activity of enzyme adenyl cyclase. This is the enzyme of the human cell. Adenyl cyclase produces a large amount of cyclic adenosine monophosphate, and this is a mediator of opening of the poring systems that of the pores that release from the cell sodium, water, chlorine ions, and other ions. Here you see the same Vibrio cholera toxin activity. A and B subunits. B subunits specific reacts to the gangliosides on the surface of the target cells, the epithelial cells of the small intestine. After that, A portion becomes liberated from the B, contact to the so-called G protein. G protein activates adenyl cyclase. Adenyl cyclase causes accumulation in the cell of cyclic adenosine monophosphate. This monophosphate activates one of the specific protein kinases, the regulatory enzymes that cause the releasing via the specific pore instruments of chlorine, sodium, and water. And the cell loses water, loses ions, and quickly dies. The next, Clostridium tetani exotoxin. Clostridium tetani produce exotoxin that is named tetanospasmin. Tetana because tetany, aspasmin causing spasm of the muscles. This toxin enters to the human body, usually from the infected place when Clostridium tetani replicating the place in the wound of the patient. And what is happening in the synapses? Tetanospasmin associated to the postsynaptic membrane. This is one nerve fiber, this is the second. When nerve impulse comes to this part of the nerve ending, acetylcholine is released and transmits signal causing contraction of muscles. But this is a excitatory transmitters release is unchanged, but inhibitory transmitters release is changed, is inhibited. What are these other alternative neurotransmitters? Gamma-aminobutyric acid. It means that synapses, nerve synapses, when nerve impulse is transmitted via gamma-aminobutyric acid, can act because presynaptic membrane of this synapses is blocked by the tetanospasmin that prevents the release of the gamma-aminobutyric acid to the synaptic cleft, and nerve impulse, the inhibited impulse, can be realized. Muscles are contracted but can be relaxed. But this very long uncontrolled spasm is the typical symptom of the tetanus. The next, Clostridium botulinum. The next bacterium, Clostridium botulinum. Botulinum, this is a food poisoning. Usually toxin occurs in the food, canned meat or canned mushrooms, for example, and during the eating enters to the stomach, and after to the small intestine becomes absorbed from intestine to the blood. So botulinic toxin, also AB subunit toxin, enters to the synapse. This is a neuromuscular synapse. In the upper part you see nerve ending, in the lower you see the muscular fiber. So presynaptic membrane on the side of the neuron, is coated by the botulinic toxin and prevents release when nerve impulse enters to this part of the neuron. Vesicles with the acetylcholine try to be released in the synaptic cleft, but it becomes impossible due to the toxin. Acetylcholine is not can't maintain transmission of signal from nerve ending to the muscle, and muscle remains relaxed. So botulinum, Clostridium botulinum causing disease that is demonstrated by relaxation of muscle, because motor neuron can realize passage of electric signal in the form of the transform in the form into chemical signal in the form of the acetylcholine, and in this case neuromuscular reaction is absent. Here you see a more detailed scheme of this botulinic agent. What now we see about neuromuscular synapse. Well, this is a synaptic cleft, and this is synaptic cleft. This is a presynaptic membrane, and postsynaptic membrane. So what is happening in the normal conditions? Synaptic vesicles contain acetylcholine. When nerve impulse enters to this place, this nerve impulse stimulate interaction of so-called snare proteins, synabrevin, SNAP25 with syntaxin. They interact one to other, forming the complex of proteins that attach the vesicle contain acetylcholine to the presynaptic membrane, or the fusing of this vesicle to the membrane, release of the acetylcholine to the synaptic cleft, and through the synaptic nerve, the signal is transported to the special receptors that, when activated, stimulate contraction of muscles. What is happened when endotoxin through the blood enters to the synapse? So botulinic toxin enters to the synaptic vesicles, and if nerve signal comes here, with this botulinic toxin stimulates not the aggregation of the snare proteins, but destroying of snare proteins. And they can establish contact of the synaptic vesicles with the acetylcholine to the fusion of these vesicles to the presynaptic membrane. Of course, lesion of acetylcholine in the synaptic cleft is absent, and nerve signal is not transported. No result, this is a relaxation of muscles and capacity of contracting of these muscles. A few words about endotoxins. Endotoxins are integral part of the outer membrane, or this outer membrane, of the cell wall of gram-negative bacteria. You know that cell wall of gram-negative bacteria consists of the plasma membrane, small layer of the peptidoglycan, and outer membrane. Outer membrane consists of phospholipids and external layer of the lipopolysaccharide. Every molecule of lipopolysaccharide consists of the polysaccharide side chains, polysaccharide core, and two molecules of the fatty acid. Well, this lipid is named lipid A, and this lipid A has the toxic capacity. When cell dies, outer membrane is disrupted, lipopolysaccharide molecules are released to the outside, and this polysaccharide lipid A interacts with the phagocytotic cells and endocytotic cells, and they release two necrotic factor, interleukin-1, that initiate changing of vascular permeability, exchange of the gases, and finally inflammation and in some cases death. Endotoxic mediated properties, clinical properties: fever, leukopenia followed by leukocytosis, activation of complement, this is a protective immune protein system, thrombopenia, disseminated intravascular coagulation, decreased peripheral circulation and perfusion of major organs, shock, this is a fall of the blood pressure, that is followed by death. Sources of infection. Sources of infection are different. Human beings, animals, insects, soil, water, and food. Human beings. The parasite microbe may originate from a patient or carrier. Patient, this is a person who demonstrates symptoms. Carrier, this is an infected person that doesn't demonstrate symptoms of disease, but has bacterium and releases the bacteria to the outside. Mode of transmission of infection caused by such mechanism is human to human, And infection is now named anthropootic if human to human transmission is happening, anthroponosis. Animals. Infectious diseases transferred from animals are called zoonosis. Anthroponosis disease that spreads among humans, zoonosis spreads among the animals. Animals may serve as the reservoir, natural reservoir, of human infections when maintaining the parasites in the nature. Insects. Some microbes may cause arthropod-borne diseases. Arthropod-borne diseases that are transmitted by blood-sucking insects. So these microbes may cause arthropod-borne diseases because these pathogens may be transmitted to human by blood-sucking insects. Mosquitoes, ticks, lice, what here you see, lice filled with the blood of human, mice, fleas, flies. All these insects, blood-sucking insects that may transfer disease from human to human, from human to animal, from animal to human by the biting, all these insects are named vectors. And infection of this type is named vector infection. Soil. Some pathogens are able of surviving the soil for a long period of time. Water may act as a source of infection due to the contamination with such pathogens as the Vibrio cholera. Soil, for example, this is a place where may replicate in the warm climate Clostridium tetani, causing tetanus. I told you about tetanus toxin before. Vibrio cholera also we said about tried to characterize mechanism activity of Vibrio cholera exotoxins. But for Clostridium tetani, Vibrio cholera exotoxins are the main, most important valence factors. Food as a source of infection. The presence of pathogen in food may be due to the external contamination, poisoning by Staphylococcus aureus, or due to the pre-existing infection in animal products. By the way, we tried to characterize before mechanism of activity of the botulinic toxin. Botulinic toxin, this is a major virus factor of botulism. Botulism, this is a food-borne disease because it enters through the mouth to the human body. In this slide, you see insects, different, and snails. Then we act as vectors. Vectors in the infectious diseases. Some ticks, flies, for example, lice, snails. Infection may be maintained in such insects and snails by transaviral or transstadial passage. It means transaviral from the maternal tick to the mite, to the daughter, so larval type of the tick. It means different in the different ages, these parasites, they change their shape and size, but becomes infected by the microbes. Stages of infectious diseases. What if we'll try to analyze how disease develops with the time and how it's changed the number of infectious agents in the human body? We have to subdivide all the disease development of this period to the different periods. First period in the development of the infectious disease is incubation period. What here? You see that green line demonstrating concentration of microbes in the body, the concentration is low. It means concentration of bacteria is low, infection is initiated, but signs and symptoms of disease are absent. The next period is prodromal. Vague symptoms appear. It means non-specific symptoms because number of bacteria in the human body is increased. When this number is increased very intensively, what you see here, this logarithmic accumulation of bacteria, invasive phase of the disease is happening. Most severe signs and symptoms are typical here. After that, ECMA demonstrates the maximum of the concentration of bacteria, maximum in the appearance of symptoms. After ECMA, if person survives, usually what is this phase, immune reactions are developed and start to influence to the lowering of the concentration of microbes in the human body. Some antibodies appear and specific lymphocytes, and due to this pressure of immune system, bacterial population is lowered, bacterial concentration becomes low, period of disease is named decline phase. Decline signs and symptoms happen. They're weaker than what most severe in the invasive phase of the disease. And finally, convalescent period started. Convalescent period that is characterized by the disappearance of microbes. This is about one illustration. Second, illustration of the same things. Vertical axis demonstrates severity of illness and replication of pathogens. Peculiarity of this figure is here is shown so-called clinical threshold. This is a minimum concentration of the bacteria in the human organism that associated to appearance of the clinical symptoms. If concentration of bacteria in the human body is changed, but all these changes at the lower level than clinical threshold, so-called subclinical diseases develop. It means incubation period, prodromal period, ACMA, convalescent period, clinical, decline periods. They are not visible on the level of the clinical symptoms because common concentration of bacteria, even in the ACMA period, in this infection, in this case, becomes not enough to cause the clinical manifestations. It means, what this dotted red line demonstrate subclinical disease development and absence of the achievement of the threshold clinical. If clinical threshold is achieved, incubation period clinical manifestations happen prodromal when the certain period of the prodromal infection symptoms appear because number of bacteria is higher than clinical threshold. After that, specific invasive phase of the disease happen. In the invasive phase, patient may die if process becomes very severe. After that, decline phases happen, and here you see typical decline phase with healing and development of chronic disease. Chronic disease is a long-lasting disease where bacteria don't disappear completely, remain in the human body and from time to time cause the increasing of the symptoms of the disease. Portals of entry and portals of exit. Portal of entry, this is the organ so that microbe may enter to the human body causing disease. Portal of exit, this is the organ so that microbe may be released to the environment from the patient's organism. But mouth, this is a portal of entry, what's green line, and portal of exit, blue line. It means mouth, through the mouth with the food, with the respiratory droplets, some kissing materials, microbes may enter to the human body, but in the same time through the mouth it may be released. Through the mouth, microbes may enter to the lungs, to the respiratory system, and through the respiratory system also may be released the portal of the exit. So urogenital tract also may serve as the portal of entry and portal of the exit of infection. Anus, large intestine, usually the portal of the exit only. Eyes may serve as the portal of entry and portal of exit with the infected tears. Direct inoculation may happen during the injuring of tissues, and arthropod-borne organisms, here mosquitoes may inoculate pathogens directly to the blood in the period of the cycling of the blood. No arthropods serve as the vectors in this case. This is example of vector infection. Modes of transmission. Modes of transmission of infection may be different. Contact, first of all, that may be direct and indirect. Infection may be acquired by direct or indirect contact. Direct contact is the method of the spreading of the disease by shaking hands, kissing, touching, or having sexual contact. What? Shaking hands, kissing. Some infections are named kissing disease. Some of the herpes viruses are named the kissing diseases. Sexual contact also is the example of direct contact. Indirect contact is the method of the direct contact, method of transmission of infection through the agency of the fomites. Fomites, they are different things that may be infected by the one person, taken to the other person, and through this fomite, other person also may acquire the disease. Fomites, for example, clothing, pencils, face towels, and toys. What here you see this toy may be contaminated by the saliva of this baby. If baby is infected, if this baby gives this to the other, some child may serve as the fomite transmitting agent from one person to other. The fomites may be contaminated by a pathogen from one person and act as the vehicle for the pathogen transmitted to another person. Other modes of the transmission: inhalation, finger-borne infections, and injection. Inhalation of a pathogen is a very typical method of transmission of the respiratory infections. For example, influenza, tuberculosis. Here you see what is happened in the moment of the coughing or sneezing. Some millions of the small droplets of the saliva infected by the microbes are released outside. But remember this slide, please, and pay attention to the mask bearing, where we say about prevention of such disease as the coronavirus infection. Pathogens resistant to drying may remain viable in secretions for a long period of time. Secretions desiccate and becomes part of the dust that also may be infected, and also may be inhaled, and this dust may serve as the transmitter of the disease. Finger-borne infections and finger-borne transmission may be recognized in the case of pathogen entering either through the mouth or through the nose and eyes. But coronavirus infection is a typical finger-borne infection. It means that not only mask wearing is recommended to prevent spread of this disease by treatment of the skin of the hands by the disinfectants containing alcohol and detergents. Ingestion. The mode of transmission is very important. Ingestion of food or greens contaminated by the pathogens may cause intestinal infections, waterborne infections, cholera example, foodborne infections, food poisoning, for example, botulism, this is also one type of food poisoning, or hand-borne infections, dysentery. Inoculation, the next mode of transmission infection. Some pathogens, when inoculated directly in the tissues of the host, can cause infectious diseases. Example, tetanus. Tetanus spores implanted in the depth of wound from contaminated rusty nails. It means rusty nails and wound caused by rusty nails is the typical mechanism of the acquiring of the tetanus. The pathogens inoculated may cause the so-called iatrogenic infections. Iatrogenic infections, when sterile medical equipment, for example, syringes or transfusion of affected blood, are employed. This way, hepatitis B, hepatitis C virus infection is transmitted, human immunodeficiency virus infection also. Congenital or vertical transmission. Congenital or vertical transmission is known as the ability of some pathogen to cross the placenta barrier and infect the fetus in utero. This may result in abortion, miscarriage, stillbirth. Live infants may be born with a manifestation of a disease. For example, congenital syphilis, Zika virus infection. Well, here in this drawing, you see pregnant woman infected with the Zika virus. This is one of the arbovirus infections transmitted by arthropods, by mosquitoes. Mosquitoes bite pregnant woman, and through the blood of the woman, these viruses may pass through the placenta, infecting brain of the patients and may cause ocular abnormalities, microcephaly, very small brain, long-term outcome unknown, fetal demise, and other defects. Teratogenic effects, such as intrauterine infection with the rubella virus, may interfere with agonogenesis and lead to congenital malformation. Congenital malformation, this is a normal development of the organs of the fetus, first of all including brain. Classification of infections. Now I'll try to comment some terms. For example, primary infection. Primary infection is an infection is an initial infection with a parasite in a previously healthy host. This infection caused by one parasite. Previously healthy host person is infected. One parasite causing disease. Second term, reinfection, repeated infection. Reinfection is the subsequent infection by the same parasite in the host. Reinfection means that a certain parasite caused disease time before, 1 year before. For example, gonorrhea, sexually transmitted disease. After that, patient was treated, and this parasite was eliminated from the human body. But if after that, after 1 year, the same patient again becomes infected by the same parasite, this disease appears again. But before first episode of disease and second episode of disease, we saw the period free from parasite. It means patients didn't have parasite is in his body. Reinfection, repeating infection. Third, secondary infection. But in this case, we say about more than one pathogen. Second infection is a new infection with a new parasite. It means parasite number 2. In a host whose resistance is lowered by the pre-existing infectious disease caused by primary pathogen. It means pathogen number 1. What is happening in the second infection? Pathogen number 1, for example, human immunodeficiency virus, infects patient and cause destroying of immune system. In these conditions, parasite number in U, this may be fungus, Candida albicans, for example, or herpes virus, consumes this situation where patient has lowered immunity and colonizes tissues of this organism. Without primary parasite, this parasite number 2 can cause disease. But when parasite number 1 debilitates, causing debilitation of immune system, parasite number 2 requires the chance for disease development. The next, option 4, cross or mixed infection. Cross or mixed infection is a new infection, number 2, caused by pathogen number 2, in a patient already suffering from a disease caused by primary pathogen, number 1. Both parasites infect the host simultaneously. What is the difference between secondary infection and cross infection? In both cases, what is common? In both cases, we say about 2 microorganisms. In the mixed cross infection, we say that both first and second parasite occur at the same time. In the second infection, parasite number 1 and parasite number 2 may occur in different periods of life of this patient. But in principle, cross infection, this is a variant of secondary infection. Option number 5, next. Term is nosocomial infection. Nosocomial infection is the cross infection occurring in hospitals. Cross infection when parasite number 1 causes disease together with parasite number 2. But if this situation is happening in the hospital, infection acquired in the hospital is named nosocomial infection. Classification depending on the classification infections depending on the source of infections are classified as endogenous and exogenous infection. Endogenous infection is where the source of infection is the host's own body. For example, in our body occur Candida albicans, and if this Candida colonizes the tissues and causes disease, this would be a typical endogenous infection. Exogenous infection is the infection where the source of infection is from external source. For example, Ivory Coast Ebola virus is the typical parasite of the chimpanzees. This Ivory Coast Ebola virus can be passed to humans from infected chimpanzees. The next classification of infections, depending on localization of the process. Depending on localization, infections are subdivided to local, focal, and generalized. Local infections is confined to the specific area of the body. For example, this whitlow that you see in the finger, this is herpetic infection that is happening only within this place of the body. This would be local infection. The second option, focal infection, focus is a local process, but pathogen or toxin of pathogen can spread to other area. If this whitlow will spread to the other area of these fingers and other fingers of this and other fingers, this process may be named the focal. Local spreading of the disease is happening. And generalized infection. Generalized is a disseminated process when pathogen is distributed in many organs, usually distributed via blood. It means bacteremia, or toxemia. This is a condition required for formation of generalized infection. Let's continue. Depending on the clinical effects of infection, that may be classified into inapparent subclinical infection as atypical infection. Inapparent infection or subclinical infection, you remember this figure. Subclinical and inapparent, this is a disease, is not demonstrated clinically, but may be registered by laboratory examination. Condition is happen when total concentration of bacteria in the human body is less than clinical threshold of the development of the disease. Inapparent or subclinical infection is the one where clinical effects are not apparent. People with an apparent infection can spread the disease to others without demonstrating symptoms. Atypical infection, second type, is one in which the typical or characteristic clinical manifestation of the particular infectious disease are not present. In a typical infection, typical clinical manifestations are present, but non-typical, for example, moderate fever, may be present. Is an apparent infection, no symptoms occur, but is the atypical, they occur. But these symptoms are not, cannot be used to establish diagnosis. Too common, too unclear, but we understand that individual is sick. Depending on the time of development of the disease, infectious disease may be subdivided to the acute and chronic disease. An acute disease is characterized by sudden onset and a short course. Short course of the disease, usually less than 3 months, with the typical clinical manifestation. Ebola virus infection is shown here in this photograph. The typical distinctive characteristic of this disease are bleedings from the all body openings, what here from the nose and from the mouth. Chronic diseases. Chronic disease develops more slowly, less severe, and lasts for a longer period of time, usually more than 6 months. Clinical manifestations may be present or may not, because in the different periods of this long-lasting disease, conditions organism becomes different. Chronic disease may be characterized by so-called relapse. Relapse is the return of the symptoms of the same disease. Example is relapsing fever, paratyphoid fever. But here you see figure. When in the horizontal line you see weeks of the disease development. In the vertical temperature, body temperature. You see episodes of the high body temperature, but here you see in the upper part 3 episodes, in the lower part 4 episodes. You see that during the first week, temperature remains normal. After that, very quickly, before the second week, it's increased up to the 40 Celsius. 1 or 2 days after that, is normalized again for this first relapse. After the second relapse has happened, after the third, and the analogous situation you see in the case number 2, but number of relapses is higher. It means relapse is the return of the symptoms. Some diseases are named relapsing fevers because they have the typical timetable of the changing of the clinical manifestations and periods of the fever. And in the contrast to the acute and chronic diseases, subacute diseases may occur. Subacute is the intermediate disease between an acute and chronic disease that lasts from 3 to 6 months. It means if disease lasts lower than 3 months, this is acute, more than 6 months, this is a chronic, and subacute when disease lasts from 3 to 6 months usually. Some terms: latent disease, carrier state, and carrier state again. Latent disease is disease during which the causative agent for a long period of time can be found in host tissue or organ, not causing clinically marked reaction of the host. It means latent disease characterized by presence of causative agent of the disease during a long time, but clinically is not manifested. Carrier state is the form of the infectious disease during which the causative agent for a long time may be isolated from the body of the infected person without clinical manifestation of the disease. It means in the carrier state, bacterium present in the human body doesn't cause clinical manifestation and may be detected by laboratory tests. Carrier state with a duration of less than 6 months is named acute. Carrier state with a duration of more than 6 months is named chronic. Now, this is the last slide. Thank you for your interest for the lecture. I hope this lecture will help you to recognize many medical terms correctly in the future and to react adequately to these principles and terms that applied for infections. Thank you. Goodbye. Понимаете, я сейчас закончил эту лекцию, но она должна быть запущена буквально через полчаса. Когда закончил, нужно ее записать. Я еще с ней не делал. Просто записать и в 15:40 ее выставить.